Hello, everyone, and welcome back to the NWR Virtual Healthcare Day for November 2021. My name is Finn Blake, and I will be your host for the day. Up next this morning, we have Chimeric Therapeutics. Chimeric is a clinical stage cell therapy company and the ASX leader in cell therapy, focused on bringing the promise of cell therapy to life for more patients with cancer. Chimeric is currently in a phase 1 clinical trial for its lead asset, CLTX, CAR-T, a novel and promising CAR-T therapy for the treatment of patients with GBM, with the first data from a trial that was recently announced. The company has also secured a second asset in its portfolio, having an exclusive license to a novel CAR-T for solid tumors from renowned cell therapy institution, the University of Pennsylvania. Joining me this morning will be Jennifer Chow, the CEO. Jennifer joined Chimeric from Kite Pharma, one of the leading players in cell therapy worldwide, where she was head of global marketing, commercial operations, and analytics. She also has spent time at Celgene and Roche during her career. I will just say before we get started that if anyone in the audience would like to submit a question for the Q&A session at the end of the presentation, please do type it in using the Q&A panel on your screen at any time. Without further ado, I will hand it straight over to Jennifer. Go ahead.
Perfect. Thank you so much, Finn. And thank you so much for having me join you today. It's a real pleasure. I appreciate the opportunity. Thank you, everybody, for joining and letting me talk to you and introduce you or update you on Chimeric. For those I have not yet been in front of, my name is Jennifer Chow, and I'm the CEO of Chimeric. So just a quick highlight in terms of a corporate snapshot for Chimeric. As Finn said, we're very proud to be the ASX leader in cell therapy as the only clinical stage cell therapy company in Australia. And that clinical stage asset is our lead asset, CHM 1101, or what we call our chlorotoxin CAR-T. And that asset is the asset also that Finn was talking about, having some very early stage clinical data that has just come hot off the wire last week. Our second asset we licensed in July from the University of Pennsylvania, and that is CHM 2101, or our CDH17 CAR-T. And we're very excited as we're making very rapid progress to market or to a clinical trial with that asset. We continue to further advance the development of our pipeline and really are looking to bring in additional novel cell therapies. And we're doing that with a team that I'm incredibly proud of. They're an industry-leading team in cell therapy development and certainly have the experience and the expertise to drive all of our programs forward. Just a little bit of history. We were founded in 2020. We listed on the ASX under the symbol CHM in January of 2021 with an oversubscribed raise of $35 million. And we truly appreciate all of the support that we've had since then. Our current market cap is around $100 million. And at the end of September, we had about $18 million cash on hand. We have 330 million shares on issue, and board and management ownership accounts for about 30% of that. So what are we all about? Our mission, as Finn said, is to bring the promise of cell therapy to life. But what does that really kind of mean? What it means to us is we really are looking to find cures for cancer. We've seen with cell therapies potential cures for patients, and you see highlighted on the left and the right of these screens patient scans where CAR-T therapies or cell therapies have been given, and you can see that the tumors have completely melted away and disappeared. And on the right, a patient, Damas Padilla, who I have the honor of knowing, who actually now 5 and 6 years after he thought that he had run out of options to address his lymphoma, is now continuing to enjoy his life cancer-free. So that is the promise that cell therapy can bring to patients with cancer, the promise of curative intent. And our mission is really to make sure that we bring that forward to more people around the world. The reason that cell therapy investment landscape looks like this and is the most active landscape in biotech today is because of people looking for those curative therapies. And so in 2020 alone, we saw over $20 billion of investment in this space. And what we've really seen in the last 18 to 24 months is a very strong commitment on behalf of all of the big pharmaceutical companies to move in and invest in this space. through acquisitions. You see some of the acquisitions that, by large companies highlighted here, the Gilead acquisition of Kite for $12 billion and the Celgene acquisition of Juno for $9 billion. And through partnerships. So you certainly see companies, well-known companies like Janssen, Eli Lilly, Merck, Roche, all moving into this space, partnering with smaller cell therapy companies for 1, 2, 3 assets at very high partnership milestones and royalty payments. So really, this continues to be an incredibly active investment space. For us, when we think about how do we actually bring that mission that we have to life, it's first and foremost centers around the assets that we bring into the company for development. And so this is how we really strategically think about the assets as we're evaluating opportunities. We look for assets that are cell therapies in oncology, and then we look to see whether there are novel designs. So is there something unique about them? And then we look at the type of cells they use. So a lot of the cell therapies that currently are in development use T cells. So we started to look at both T cells and alternative cell types like NK cells or macrophages. We also look at alternatives around cell sources. Again, the therapies that are in development and definitely all of the therapies that are commercially approved currently are individualized, or what we call autologous therapies, meaning that we use a patient's own blood to be able to give them their own therapy. With off-the-shelf, so the alternative cell source is an off-the-shelf, or what we refer to as an allogeneic cell source. That's when we actually use a healthy donor who's willing to give their blood, and we engineer that blood to develop a therapy for other people. So as we build out our portfolio, this is really the strategic framework in which we've been using. Right now, as I mentioned, we have 2 assets in our portfolio. They're both novel designs, they both utilize T cells, and they're both individualized therapies. The first is our chlortoxin CAR-T, which is an ongoing phase 1 clinical trial, and I'll show you some of the very early data from that trial. And that asset was licensed by Chimeric from City of Hope Hospital in California. The second asset that we brought in July is our CDH17 CAR-T that we brought in from the University of Pennsylvania and plan to move into a phase 1 clinical trial next year. Now, for those that aren't familiar with what CAR-T cell therapy is, here's just a bit of an explanation around what a CAR-T cell therapy actually is. So cell therapy itself, at its simplest sort of description or definition, is the transfer of live cells into a patient to treat or cure a disease. And if you think about it, we're all very familiar with cell therapy because we've all heard or known of a blood transfusion. And a blood transfusion is the simplest form or example of a cell therapy. CAR-T cell therapies are just a little bit amped up because we engineer them genetically. So what you see on the left-hand side is a bit of a diagram or a schematic of what happens in terms of how we make these therapies. Essentially what we do is we take a patient who has a specific type of cancer and we take blood from them. We ship that blood off to a manufacturing facility where that patient's own cells are engineered with a chimeric antigen receptor. And then we ship those cells back, those engineered cells back to the patient, and they're infused. On the right-hand side, you see what happens when the cells are infused into a patient. Essentially, from that genetic engineering that we did in the manufacturing facility, you now see this little chimeric antigen receptor that gets expressed on the outside of the cell. And what that does is it goes seeking a very specific type of cancer cell. And when it finds that cancer cell, it attaches to the cancer cell and sends a message to kill the cell. And so the cell winds up being killed. So this is really, both of our therapies currently utilize this technology and this process for manufacturing and how they work once they're infused inside a patient. Both of our assets right now are moving into different disease areas. We have the one ongoing phase 1 clinical trial in glioblastoma, and then we have 2 additional clinical trials planned for 2022. So by the end of 2022, we'll have 3 clinical trials ongoing. Importantly, we're also looking across 7 different disease areas. So when we really think forward about the types of patients we can help, what we're targeting is patients in 7 different disease areas to be able to help just with these 2 therapies alone. Now, I'll start with giving a little bit more detail about our lead asset, which is the one that's in the phase 1 clinical trial for glioblastoma, our chlortoxin CAR-T. Our chlortoxin CAR-T was designed and studied preclinically to address glioblastoma. Glioblastoma is a type of brain cancer, and for anybody that's ever had to come in contact with it, they know it's one of the most lethal types of cancer out there. Unfortunately, patients that are diagnosed with glioblastoma have a very limited life expectancy, usually around 1 year, and very few survive into 2 or 3 years. So certainly in this disease area, you can see that there was a need for much better therapies to help these patients. Our chlortoxin CAR-T therapy utilizes this very unique chlortoxin tumor targeting domain. And so while chlortoxin has been used safely in patients for a long time to actually highlight tumor cells as an imaging agent, this is the first time that it's actually been used to highlight the cancer cells and attach as a CAR-T therapy. What we were able to see in the preclinical models before we actually took this asset to clinic was chlortoxin is able to find glioblastoma cells, to attach itself to those cells and send that message to kill those cells better than other immunotherapies out there. So really great preclinical evidence is what had us take this asset to the clinic. We actually hold a global exclusive license to that CAR-T, chlortoxin CAR-T therapy, and we do have robust and broad IP around this through to 2036, so very comfortable with the IP protection. This is the schematic of the current phase 1 clinical trial. So in all phase 1 clinical trials, the primary objective is safety. So we really, because this is the first time we've moved it into patients, we want to make sure that it is safe for people. And then what we're looking at is to determine what is the right dose to give to people. So how do we optimize the dose to get the best possible efficacy with the least possible side effects? So that's really what we're looking for as well. We also look for additional insight into how the drug is behaving or how the CAR-T is behaving. So we look for efficacy measures and what we call bioactivity measures, which is really about how are the cells behaving when they're infused into a patient. This particular trial was designed with 4 different dose levels that you can see laid out here. On the far left is dose level 1. And so dose level 1 started by giving patients a very low dose of 44 million cells. And we give such a low dose of cells when we first go into a dose level or into the first dose level, because this is the first time, again, it's been in patients and we want to make sure and protect patient safety above all else. You can see that by the time we get to dose level 4 on the far right, we actually increase the dose of cells that we're giving patients tenfold. So we're giving 440 million cells in dose level 4. The other thing about this particular asset that's pretty unique is the way that we actually give the CAR-T cells. So in dose level 1, what you see there is this image of a patient's brain and what kind of looks like a needle sticking out of it. And that's actually how we administer the cells. Patients have surgery. We insert a catheter into the tumor cavity. And then what we do is we directly administer the cells in through that catheter directly into the tumor cavity. In dose level 1, that was the one way we use that one way to administer cells to patients. As we move to dose level 2, and then also as we continue with dose level 3 and 4, we'll continue to use the one intratumoral catheter to give patient cells directly into the tumor cavity, but we've added a second catheter. And you're going to see a little bit of evidence about why this second catheter is so important. We've added the second catheter to allow intraventricular access and administration, because we believed when we designed the trial that this would give patients a better opportunity to have all of their glioblastoma affected or impacted, killed. So the trial completed dose level 1. What we saw over the course of the last week or so was the very early evidence of dose level 1. So now that is only 4 patients, so this is very early data, but certainly we were incredibly encouraged by the data that we saw coming out of those dose level 1 patients. And I'll just highlight some of the key things that we saw. Really, we safety, efficacy, and bioactivity. So from a safety perspective, Very importantly, we saw no dose-limiting toxicities, so no safety events or adverse events that were severe enough that would make us stop the trial at all. We saw that generally the toxin CAR T therapy was well tolerated by all patients, so that was very encouraging. What we did note is that there was one grade 3 cerebral edema, but the clinician attributed it to only possibly being as a result of the chlorotoxin CAR T cells. So when there's an event or an adverse event that's a grade 3 event like this one was, what happens is the clinician is asked, is it possible, is it probable, or is it very, absolute that it was the drug, in this case, the chlorotoxin CAR T cells, that caused the event? And so in this particular situation, the clinician said that he felt it was only possible that the chlorotoxin CAR T cells caused the event, and recognized that, and he actually recognized that glioblastoma patients, unfortunately, see cerebral edema as a common adverse event. So promising safety, we'll certainly keep our eye on all of these things, but we really felt that this was promising safety for that first dose cohort. So moving from safety to efficacy, just to give you a sense of what we saw from an efficacy perspective. Now, again, only 4 patients, so very early days, but we saw 75% disease control in that very low dose of cells. We honestly did not expect to see very much because we were recognizing that we were only giving patients 44 million cells. So here, 3 out of the 4 patients that were treated with the chlorotoxin CAR T achieved an overall best response of stable disease, meaning that their disease did not progress, it remained stable, so it was in control over the course of time. And we saw that that course of time could last up to 8 weeks, which was very positive for us as an early signal at a low dose in this trial. Just to give you a sense of how we think about that in relation to, wow, well, it doesn't sound, 8 weeks doesn't sound like much. First of all, I'll remind you, it's a very low dose that we're giving patients here. We're going to get to 10 times that dose level. But I also thought might help to get a bit of a sense of what are the analogs out there. So what you see here is a list of different therapies that are actually being developed in glioblastoma in the same disease as the chlorotoxin CAR T. What you can see across the phase 2 and even the phase 1 trials is disease control rates, even when people are using full dose of other therapies. So all of these phase 2s would be using their full dose of therapy that are somewhere between 25 and maybe 50%. So that disease control rate of 75% that we saw in our early patients is very promising. You also see a median progression-free survival that very sadly for these patients ranges from just over 1 month to just under 3 months. So in terms of a patient's ability to hold that response, unfortunately, what we've seen is that there hasn't been good responses. So those 8 weeks of durability we're seeing at that very low dose, again, in relation to what we expect to see with this particular disease, was very encouraging to us. I want to highlight this particular MRI scan is what you're seeing on the left-hand side, because this was something that certainly caused a great deal of excitement when it was presented at the Scientific Congress this past weekend. What you actually see there is an MRI scan of one of the patients that was treated with chlorotoxin. What you see on the top image, at the very top where the arrow is pointing, and on the second image where the arrow is pointing, is that's where the catheter was placed into the patient's brain and the cells, the chlorotoxin CAR T cells, were administered. And what we saw was that even after the 8 weeks, this particular patient had no recurrence at that place where the cells were administered in their brain. What you see in the image at the very bottom of the screen is recurrence of the disease, but recurrence in a place in the brain where the chlorotoxin CAR T cells were not administered. So where they were directly put into the patient's brain, we saw no recurrence, and we only saw the recurrence or progression where the cells were not administered. This was important, and it generated excitement because, as I was talking before about the design of the trial, we know that in dose level 1, so this was this group of patients, patients were only receiving the cells through that one catheter into the one spot in the brain. But as we've moved through dose levels 2, 3, and 4, we're actually going to be giving them cells in the 2 different catheters. So what we really hope is that what we're seeing on the left with the MRI and no recurrence where the cells were actually administered, we'll be able to impact the other part of the brain by giving the other route of administration as well. So this was really something that was very positive and gives us lots of reason for optimism as we moved into those dual routes of administration. The other thing that I talked about that we looked at in this data set was encouraging bioactivity. And what that really is important for is to assess when we infuse the cells into a patient, are the cells staying alive? So we know that we need the cells to stay alive to be able to do their job. They can't find the cancer cells, attach and kill the cancer cells if they've all died off. So what we were able to establish through liquid biopsy was that the cells were staying alive all the way through treatment. So that was something really important for us to establish and know, because it means that the cells then have the opportunity to do their job to find and kill the cancer cells. So all in all, although very early days and a very small set of patients, very encouraging early clinical data that certainly leads us to have a lot of enthusiasm and optimism as we move forward into the rest of the trial. Those early signs of efficacy, a promising safety profile, the encouraging bioactivity, and we also saw some great positive basic research insights that help us think about how we develop this asset as we move forward. So very encouraging from that data set. As I mentioned at the beginning, we're now in dose level 2, so we're continuing to advance this trial. The patients in dose level 2 are being recruited and infused now. Next year, we anticipate moving into dose level 3 and then finishing off dose level 4 in the trial as well. In August of this year, we importantly were able to get FDA clearance for our own IND, which allows us to accelerate and expand the clinical development program for chlorotoxin. This particular trial, the chlorotoxin glioblastoma trial, is currently open at 1 site in the United States. Next year, we're planning to open 2 additional clinical trial sites, which will allow us to accelerate recruitment for the trial. Additionally, we know that chlorotoxin actually has the opportunity or the potential to work for other tumor types as well. And some of that important or interesting basic science research that I talked about actually showed us that one of the disease areas that it may work very well in is metastatic melanoma. So we have a second trial planned to open in 2022, which will look at chlorotoxin in a number of other solid tumors, melanoma really being the lead indication for that trial at this time. So lots of excitement and lots of development moving forward very rapidly with our first asset here, chlorotoxin. This is our second asset in our pipeline, so CHM 2101, or our CDH17 CAR-T. And we think that this asset holds incredible promise for gastrointestinal cancers. So this particular asset we licensed from the University of Pennsylvania in July, and our team was thrilled to have actually acquired the license as it was a very competitive process. The University of Pennsylvania cell therapy team is world renowned, and certainly they have been optimizing this asset for over a decade, so there was a lot of interest. The other thing that had so much excitement in terms of this particular asset was what we saw from a preclinical efficacy perspective. The evidence that I'll show you just quickly really suggests that we see complete tumor eradication, so complete eradication of tumors with no relapse at all in the preclinical models. So really excited to be able to move this particular asset into a phase 1 clinical trial as soon as we can. And based on the efficacy that we saw in those preclinical models, we're actually planning a phase 1 clinical trial in 4 different tumor types right away. So looking to address patients with colorectal cancer, pancreatic cancer, gastric cancer, and GI neuroendocrine tumors as well. This is what I just mentioned in terms of the preclinical efficacy that we saw with this CDH17 CAR-T that really gave us so much excitement and enthusiasm to move this into the 4 different tumor types. What you see represented in the graph on the right is what would happen, if you left a tumor to grow over time. So you see the tumor volume increasing over a period of time. The yellow and the black lines are really control lines. So basically what they're telling you is if you really didn't do anything, how would the tumor just naturally grow over a period of time? The red line is an actual therapy. So it's showing you that with a certain type of therapy, so this is a second generation CAR T, you can have some impact. You can slow down the tumor growth a little bit or make it so that the tumor doesn't grow as big, but it doesn't go away. What has us so excited about this asset is that we're the green line. So our CDH17 CAR T is the green line, where you see that the tumor volume actually comes down right under zero, so it's completely eradicated, and that is held all the way through the study, and the tumor never comes back. So this really is very promising preclinical efficacy for us, and what really gives us the confidence to move into a clinical trial with so many tumor types. For this particular asset, we are getting everything ready to move into that important first clinical trial. We were pleased that we were able to very rapidly after our licensing move forward with the first milestone on the path to securing all of our technical operations and have our plasmids released. Dr. Hua, who is the inventor of this asset, and his team at his lab at the University of Pennsylvania are working very hard to advance all of the preclinical validation we need so that we can open up that trial in those 4 tumor types next year. So in the second half of next year, we'll be filing our IND to open this trial, and we'll initiate the trial at the University of Pennsylvania and then rapidly expand the trial to a number of other clinical trial sites. So past those 2 assets, knowing that we continue to want to develop and diversify our pipeline, we are continuing to look at other opportunities. And you can see from this diagram, from a strategic perspective and diversification perspective, we're starting to look at some of the alternative cell types, things like NK cells or macrophages. And we're also starting to look at things like off-the-shelf therapies. So this is a big part of how we're starting to think about our pipeline development going forward. And one of the reasons why I think we feel like we have so many great opportunities to assess and are able to really pick and choose and find something that really strategically fits our portfolio and pipeline is because of the team that we have in place. I'm incredibly proud of a very small management team, but certainly a team that brings an incredible amount of expertise and insight and experience around the development of these assets. Cell therapies certainly are very different in terms of what they need to be developed, from technical operations to their clinical development, regulatory development, and certainly have the team here that has all the experience necessary. Just across this small management team, we've actually developed over 25 cell therapies, including 4 out of the 5 approved CAR T therapies in the United States today. So really leveraging this team to actually find the right assets, determine whether they have the right level of promise, and then be able to move them into development. So with that, I'll just sum up a little bit. Again, very proud to be in that leadership position in cell therapy in Australia with the 2 assets that we have from world-renowned centers, the University of Pennsylvania and City of Hope. We have the current ongoing phase 1 clinical trial advancing to the upper dose levels now with that promising early data. We have a second chlorotoxin CAR T clinical trial planned for 2022, and our CDH17 phase 1 clinical trial planned for 2022 as well. We have attractive near-term opportunities to grow out our pipeline and believe that our industry-leading management team experienced in cell therapy certainly enables us to do all of this in a rapid and really wonderful way moving forward. We see different value realization pathways for investors, and certainly really think through opportunities for sale of the company as our assets continue to move forward, strategic partnerships, and to develop independently. So different value realization pathways highlighted as well. With that, I'd very much like to thank you for your time and your attention, and certainly invite any questions and appreciate the opportunity to speak with you all again today.
Thanks so much for joining us, Jennifer. We've probably got time for one question, which is, what is the main reason you're confident in the Chimeric business as we approach the new year?
The main reason that's a great question. I think, I don't know if I can give you one, but if I have to give you one, it's our assets, Finn. I would generally say our assets and our people, but I think the assets just have so much promise to them. Both assets have been designed by world-leading scientists at very strong CAR-T and cell therapy centers, and they had incredible preclinical efficacy. And so I really believe that we have two assets that have great potential for patients.
Amazing. Jennifer, thank you so much for your time today. Really excited to continue tracking the Chimeric business, and yeah, looking forward to seeing what's in store.
Thank you so much, Finn. Thanks for having me. It was great to talk to everybody.